HIV stands for Human Immunodeficiency Virus, and that's really the name of the virus, whereas AIDS, this other term that we know, acquired immune deficiency syndrome, is a description of the symptoms that people with HIV will eventually display. So that's where those two names come from. The AIDS describes the symptoms, and in fact that name was developed several years before people realized that it was a virus, and then when they discovered the virus they named it HIV. As you probably know, HIV is primarily spread through sexual fluids. It can also be spread through blood and from mother to child. AIDS is a major world health concern. You can see some numbers here. Over 25 million people have died since AIDS first appeared on the sort of global stage. And right now, about 1 in every 100 adults between 15 and 49 is infected worldwide. The virus specifically targets immune cells, and it will cause the destruction or death of a white blood cell called a helper T cell. And the problem for the people who have this is that those helper T cells help protect us from infections. And so when people get sick from AIDS, what they really become sick with is a bunch of different infections that their body can't fight off anymore. That people who do not have AIDS are able to fight off. So people with AIDS, for example, can end up dying from the flu or pneumonia or things that most healthy people are not going to die from. So that's, again, the real problem is that they start to just catch all these infections. So HIV is part of a family of viruses called the retroviruses. And retroviruses are really unique and different from a lot of the other viruses. Part of the reason why they're so different is that they have RNA and single-stranded RNA as their genetic material. Because they have RNA as their genetic material, that RNA is going to have to be converted to DNA in order to infect the host cell. Because, of course, human cells have DNA as the genetic material. So the HIV virus is going to have to do something that human cells never do. And that is to go from RNA to DNA. So remember, in our body, in our normal cells, the direction of information in the cell is always DNA to RNA to protein. That is what is happening in our body. But the virus needs to be able to take its RNA and make DNA out of it. So there's nothing in the host that can do this. So the virus has to come with its own special protein called reverse transcriptase. Reverse transcriptase is an enzyme that can read RNA and turn it into DNA. So an HIV particle has the RNA genome and reverse transcriptase packaged into a capsid. So it has its capsid, it has the RNA, and then it has reverse transcriptase packed inside of it. And then HIV also has a viral envelope and spike glycoproteins on the surface that are going to help it attach to the immune cells. One of the reasons why HIV has been so difficult to treat and develop drugs for is because of reverse transcriptase. It's kind of a sloppy enzyme. It makes lots of mistakes. So it very readily makes mutations. And so HIV rapidly evolves to not be treated effectively by the drugs that we make. So that's been one of the challenges with HIV. So we'll take a look at the HIV life cycle now. And so remember, it's going to follow this basic reproductive cycle that we've seen before: attachment, entry, integration, synthesis of viral components, viral assembly, and viral release. It's going to do it a little bit different from phage. So the first thing that has to happen is attachment. So we can see the spike glycoproteins on the outside of the viral envelope, and those spike glycoproteins very specifically bind to receptors on human immune cells. So you can see it's taking advantage of these receptors. These receptors are not for HIV, right? We don't have them because we are, our cells are trying to catch HIV. The virus is taking advantage of receptors that are already there and tricking the immune cell into thinking that it's something that it's not. Because what's going to happen is when the spike glycoprotein attaches to the receptor on the host cell is going to let the virus in. So that brings us to entry. In the case of HIV, the capsid enters. So there's the capsid with the genetic material inside of it. And then there's the remains of the viral envelope because it fuses with the host membrane. So the viral envelope becomes part of the host membrane through that fusion and lets in the capsid. The capsid will get chewed up by the host cell, and then that releases the viral RNA and reverse transcriptase. And so then the very first thing that happens is that viral RNA gets converted into viral DNA. So that's immediately carried out by the cell. And then it's going to have this RNA-DNA hybrid that will get corrected into double-stranded DNA. And then that double-stranded DNA will have to be integrated. So integration is required for HIV. This double-stranded DNA will get integrated into the human genome. And again, I just want to emphasize that this has to happen for HIV. It's not going to not happen. It's not optional. So once the viral DNA is integrated into the host genome, so there it is, the viral DNA has just become part of the host genome. Now it will just get read and copied by the host machinery. So now it starts to take over the host enzymes, and the host enzymes will take that viral DNA and they're going to make viral RNA. So they're going to carry out transcription. Now remember, for HIV, viral RNA is its genetic material. So some of that viral RNA is just going to become genetic material for the new viral particles. Some of that viral RNA will be translated into viral proteins. So that's what we have happening here. It's being transcribed into new viral RNA, and you can see pieces of viral RNA floating around. Some of that viral RNA is being translated, so that's making new capsid proteins, new spike glycoproteins, new reverse transcriptase. As all of these new viral materials get made, they begin to assemble. So here's capsid proteins, the viral RNA, the reverse transcriptase assembling near the membrane, putting spike glycoproteins in there. And then what happens with HIV is that it then forms it buds out of the plasma membrane. So the capsid proteins will line up, and it'll be where the spike glycoproteins are, and it pulls off a piece of the host membrane. So the viral envelope of viruses is really a stolen piece of host membrane that it's wrapped around itself as it's come out of the host cell. So it's wrapping the host membrane around itself and forming a new viral particle. So now this viral particle is released and can infect other cells in this same person or infect somebody else. So in the case of HIV, I just want to emphasize again that it uses viral RNA and viral proteins to make new viral particles. And remember, that's different from phage. Phage packaged viral DNA with viral proteins to make new viral particles. Also remember that phage broke out of the host cell and actually directly burst open the host cell. HIV is going to bud out of the host cell, so this isn't directly killing the host cell, but it is turning the host cell into an HIV machine, and that will just keep budding out more and more of these viral particles. So I just wanted to go back to this step here and tell you a little bit of an interesting story about HIV. So they noticed that Europeans, a certain segment of the European population, so a percentage of Europeans, were resistant to HIV infection. And this surprised people, and this kind of led them on a quest to try and understand why some Europeans would be resistant. And if you think about European history, there was a very, very major event in Europe that killed a lot of people, and that was the Black Death, or the bubonic plague. Estimates are that about 75% of Europeans died from the Black Death over a 200 or 300 year period. So 75% of the population dying, leaving only 25% behind, is a pretty significant effect on your population. And what they think happened was that there was a mutation in some people that made them resistant to Black Death. So that mutation caused them to be resistant to the plague. So more of the survivors had that mutation. So that mutation became more common in the European population, whereas it's relatively rare in populations that did not suffer a lot from the bubonic plague. So Europe had bubonic plague way worse than anywhere else in the world. And because of that, it enhanced that mutant. More Europeans have that mutation. Well, fast forward to the 20th century, and it turns out that that same mutation may be conferring resistance. So the mutation is in these receptors. And so what happens is people who have this mutation, the HIV can't bind to their receptor. So because it can't attach, the virus can never enter, and so they are resistant to HIV infection. And again, it's kind of this funny gift of the Black Death 800 years ago.